Okay, I think we can start now. I'm sure some people will filter in, so we'll be accommodating, but probably this is the numbers we're going to reach. So welcome everyone. Great to have you here. Great to have you guys in person and the folks who've got on the call, which, just from the look, there's maybe about 5 to 10 people. So my name's John. Probably you picked that up already. Looking forward to being with you for the next 2 months on this course and kind of taking you through this journey on an introduction to mathematical neuroscience. So I put a message on the announcements board that we got some response there. And not everyone replied. That's okay. It was only yesterday. So, but I just want to know that just as a kind of initial question before we get into the lecture, like, is everyone who is signed up on the Moodle system and kind of saw the announcement and is kind of up and running on that side of things? Just show hands. Right. So some people know. You know because you aren't signed up, or because you haven't looked yet, or another reason? Right. Yeah. So there are a few people who couldn't get on because we hit the max. So that was you. Was there anyone else who was wanting to get on? Okay. Because I think we can probably kind of add you in. That was just a kind of feature on the web page. So, but it doesn't really matter if we go over the 35 limit anyway. So I just need you guys to follow up with me kind of afterwards separately to make sure that we add you separately. We've got a few thumbs up over there. Okay, great. Yep, slides will be coming up shortly after the lecture. Thanks for the question there. Cool. Okay, so it sounds like everyone's kind of engaged or kind of in the process of getting on board with the online system. Great. I want to keep that rolling because that's going to be our communication pathway. So let's talk a little bit about this lecture and this course. So I'm John. I'm an assistant professor at U of T, primarily in psychiatry, but also medical sciences, biomedical engineering. And I work at CAMH, Center for Addiction Mental Health, which is the mental health hospital that is just a few blocks down on College Street. I've got more to say about that in a little bit. You just filter and close the door, thanks. So as I mentioned on the announcements, apologies for having to skip a week, but as you can probably tell, I'm still a little bit husky, my voice. I had absolutely zero voice last Wednesday, so it was just like physically impossible for me to give this lecture. So apologies for that, but I just happened. Hopefully I'll get all the way through here without the voice running out, but it might get a little bit kind of quieter as we go. So introduction to mathematical neuroscience. I've got a few preliminaries before we get into the meat of this lecture. The meat of this lecture is going to be 2 parts to it. One is a kind of whirlwind skim through a few highlights, just a few little bits and bobs I want to show you about the kind of things we do in computational neuroscience, kind of things we study, and things that are kind of interest to the field. So that's going to be a kind of rapid survey without too much technical depth or taking too much time, but just to kind of show you some interesting things. And then the second part of the meat of today's lecture is what you will have seen the title, which is talking about single neuron models. Okay, and we're really going to be getting into some of the guts of how we model neural systems mathematically. So that's the main part of the lecture. The preliminaries I want to go through are to do with the partly about the course in general, how we're going to run things, And well, actually just that. So firstly, I want to say something about where we are, mainly for people who are on the call and don't know for whatever reason what the deal is with the Fields Institute, but also talk a little bit about CAMH as well. So for folks on the call, and I think probably some people who are tuning in maybe when this becomes a YouTube video and so on. So we're here at the Fields Institute. This is a really fantastic institution, a really fantastic place. I think everyone who's here, you're quite lucky that you have Fields here. It's quite a special institution, in my opinion, in the sense that it's kind of focused on an interesting topic, right? Like advancing mathematical research, but it has a kind of 1 foot in and 1 foot out of the university. And that gives it it puts it in an interesting position. Let's kind of do a lot of interesting things. So I think Fields Institute is a really great thing. I mean, the architecture is also great, right? This fantastic staircase we've got out there. So that's where we are, people who are calling in. As I mentioned briefly just now as well, down the street, 2 blocks down the street, where it's kind of where my day job is, and also Parsa's day job, I'll introduce Parsa to you in a second, is this place, the Center for Addiction and Mental Health, CAMH. And at the center at CAMH, we have the Krembil Center for Neuroinformatics. This is the center where I work and where the kind of research, some of the research you'll hear about in this course and throughout the lecture kind of goes on. So I wanted to flag that. That's also where we are. And it just so happens to be literally 2 blocks, that two kind of addresses down on College Street. Now, so that's where we are. Now, why are we here? There's 2 reasons why we're here. One is for this course. So quick question, actually. I asked this in the survey. So how did people learn about this course? I think a lot of people said from the Fields Institute website or email list. So heard through Fields, show of hands. Okay, about half heard from something other than Fields. Guess that should be everyone, but like, can you shout out word of mouth? Supervisor, who? Steve, great. Okay, good. So some word of mouth. Anything other than word of mouth? Is there? Yeah. Okay, cool. So in any case, you're here, so you know about the course. What you may or may not also know about is this. So this is kind of the reason why this exists. So Fields do 2 thematic programs every year. Thematic program is kind of half a year of programming on some topic related to mathematical sciences. This year, one of the programs is on mathematics and neuroscience, and I'm co-organizing this with Jeremie Lefebvre and Maya Fraser as like a core organizing group. Maya is the kind of champion of this. She's spearheaded it, but we're really doing this together. So there's a lot of stuff that's going to be happening mainly in the second half of this year on the general topic that we're hearing about today. This course is the first item in the program. And this is still very tentative, but this is basically what we've got lined up over the next year. It's a pretty cool, exciting, diverse mix of content. Some highlights. We're going to have a special lecture series from Karina Curto, who's a real world leader in mathematical neuroscience, coming over from the US. We're going to have Carl Friston coming from the UK, giving a distinguished lecture series in the fall. We're running a brain modes workshop. This is a kind of big international conference that we're going to kind of co-locate with our Fields programming, and a bunch of other interesting stuff going on. And we're the first in the list here. So in other words, like, it doesn't end at this course if you want more. That's the main message there. And we definitely want to have young people coming along, right? The conferences and the workshops, there's going to be a lot of researchers. It's going to be a little bit more kind of high level to some extent, but everyone is welcome, no matter the level. That's always kind of the rule with these things. So definitely follow up if you're interested in more of what you're getting here. Okay, so how is this going to work? What's the practicalities? This was on the website, so hopefully you guys know this. But just to reiterate, so every lecture, we've got 9 lectures lined up. Is going to be every session, I should say, is going to consist of a lecture, kind of traditional lecture like I'm doing right now, and then a hands-on coding-based tutorial or seminar, which is going to be based on the content of the lecture and just expanding on a few key points. But the important point is kind of getting you familiar with implementing and exploring some of the things that you're hearing about conceptually in the lectures. So that's kind of how we like to roll in the kind of teaching that I do, is always have some hands-on piece to the didactic content. So we'll have that for every lecture. Some of the coding seminar content will be presented to you in the form of assignments. So these assignments will be kind of extensions of some of the coding exercises that we'll do in the second half of the lectures. Probably one for the first half of the course, one for the second half of the course. So we'll make a few available and you can choose. Haven't exactly decided which ones we're going to offer, but those will be coming out in the next few weeks as we get going. Okay. And then at the end, so final session is going to be you guys feeding back. And that's going to be also summarizing some like exploration you've done on some of the content from the coding exercises, and also some marks for engagement. Note this, so please ask questions and, you know, make yourself known on the forum and so on. We want to hear from you. That's kind of why this is up there as well. Okay, so that's how it's going to work practically. Final preliminary, who are we? We are 3 people in terms of the people who are bringing this course to you. Myself and Jeremie. So Jeremie, is Jeremie on the call? No. Okay. Well, maybe you'll listen to the recording. So Jeremie Lefebvre, as you saw, he's a co-organizer for our thematic program, my old boss as well, but also collaborator. And the content of the lecture series is kind of based off a course that he put together a few years ago with a lot of extension. So that's Jeremie's contribution. And then the other person, of course, to mention is Parza. Put your hand up, Parza. Okay, Parza is your TA. He's already put a lot of work in on the tutorials, and he'll be helping out in a variety of ways as we go, including monitoring the chat. So I see we've already got a few messages on the chat. Great, thanks a lot. Keep them coming through. Guys are on the call, and Parza is going to be monitoring for any questions, and if there's something that you think is worth kind of butting in, then do that, or we'll take some pauses as we go to give time for that. So we want to hear from you. We want to hear from you, people who are on the call and we're watching. Okay, so that's who we are. I should say, by the way, Parza, who are you? I mean, what do you're a master of engineering, I'd say, recently graduated master of engineering student.
Yeah, graduate students with John, John's lab, working on whole brain computational models, trying to model the propagation patterns of TMS. For those who work with TMSs, essentially we zap the brain and we try to model how the propagation of wave patterns happens, and to try to use those understandings to actually inform us for better treatments with TMS.
Right. Thanks a lot. And, you know, definitely ask Parza if you're interested in life as a grad student or things like that you want to hear about from someone who's a little bit further along. Okay, so that's it for the preliminaries. Okay, final one is just a little bit about me. And this is me and my work. And this, I think, will help a little bit to contextualize what you're hearing from me in terms of the talk content, because inevitably what you hear from people is like what they know to some extent. So that's, it's kind of worth knowing, basically. So I came to Canada 10 years ago, started a research group at CAMH that I mentioned 5 years ago. And the center over at CAMH, I showed you these photos, including this one from the window. This is a nice view from the KCNI 12.4. But the concept for the center is this kind of research clinic experiment cycle where we want to kind of drive medical discovery and ultimately medical new therapies with a variety of basic science input, a lot of which is data. And then this box that I'm highlighting here is theory, right? Computational neuroscience, understanding how the brain works. So this is the kind of zoomed out concept for the KCNI over at CAMH. And then within that, my group, one of the 7 research groups there, called the Whole Brain Modeling Group, what we think about and focus on, Parza gave you an example of one of the types of things that we do, is essentially the intersection of brain imaging and computational neuroscience. That's one way of understanding it. So things like building personalized brain simulations of pathologies and interventions in mental health to try and improve interpretation, understanding of data. And there's basically 2 clusters of things that we're thinking about. We're thinking about brain activity, which often is in the form of networks. So data like EEG, rhythmic activity in EEG, and sleep, and in sensory tasks. And then the other big slice of things is brain stimulation or neuromodulation, invasive and non-invasive. Parza mentioned TMS, transcranial magnetic stimulation, which is a non-invasive type of brain stimulation that's used therapeutically for treating depression. So trying to understand the kind of neurophysiology of brain stimulation using insights from neurophysiology about brain network dynamics and activity. That's the name of the game in my research team. So some of the things we'll hear about over the course of the lecture series, I mean, I'll say when this is our stuff, but I think both in terms of the applications and in terms of the elements of the modeling approaches, there's a lot of the content we're going to talk about is something that we're working on actively. Okay, so our syllabus for the next few weeks and months is like so. You will have seen this. This was on the website list. There's an additional kind of lecture 0 that I added in at the top here, I mentioned earlier. So our main lecture today is going to be digging in on single neuron models, but I added in this kind of overview thing because I wanted to, as I mentioned, I want to give you a little bit of a kind of global picture before we get into some nuts and bolts. So that's what I want to jump in now for the next 15 to 30 minutes or so. So kind of whistle-stop tour overview of computational neuroscience as a field, as I see it, or a primer. So let's, I want to talk about 3 things. Like what is computational neuroscience in general? What is this field? And then these are 3 different ways of thinking about or say describing the kind of things that people think about who work in this field. And they're not mutually exclusive. So the first thing to say is, okay, I'm talking about computational neuroscience here. The title for the lecture series is mathematical neuroscience. I'm using those interchangeably here. Broadly speaking, those are fairly interchangeable terms. People can complain about that, but the history of the phrase computational neuroscience is kind of curious, really. I mean, it goes back to like the 50s or so, when doing anything with an equation was considered computational, or just doing anything numerical was thought of as computational. These days, like everyone who works in science, and to be honest, everyone who works in almost any industry is doing computational work one way or another. So kind of unique aspect of mathematical neuroscience that kind of gave it the term computational is a bit of a kind of anachronism. On the other hand, mathematical neuroscience is a little bit broad as well. But I'm using those interchangeably. And I think that's the right way to think about those words when you hear them. So you know, computational equals mathematical neuroscience. Now, in terms of this field, it's a very broad field, right? Because it's kind of like, it's basically neuroscience plus. It's like everything in neuroscience and then a lot of other things. It's like elements of cognitive science, computer science, physics, biophysics, chemistry, you know, psychology. So I think computational neuroscience is a huge field. But broadly speaking, there's two halves of it, two broad types of things that people study in computational neuroscience. They're either asking things about, broadly speaking, cognition, and just some kind of synonyms or related terms here, right? Like information processing, coding, representation, like complex thought, those kind of things. So you're either basically thinking about cognition, or you're thinking about, broadly speaking, what we call dynamics, or maybe we could call this physiology, where and physiology, we might think about physiology at the kind of micro scale or small spatial scale, and we're talking about cells, which we're going to be mainly focusing on today. But then all the way up to things like, you know, large networks and kind of emergent behavior that's still in the broadly the realm of like physiological dynamics. So these are the two types of things, you know, first like principal components of computational neuroscience as a field, essentially. And you'll see I've added a little dotted line down the middle here, which is indicating doing both of these is very hard. Or put another way, like finding the relationship between things we can understand in terms of dynamics and things we can understand in relation to thought and information processing is kind of the holy grail of computational neuroscience. And there's some of the things we'll talk about in this lecture series will be kind of at this interface, but broadly speaking, you're either here or you're here. And there's interesting and challenging work to do everywhere in this map. Few quick comments on modeling, like what is modeling and why do we do it? Well, the first thing that's just super important to understand from the point of view of any modeling work is that we're always making abstractions and idealizations and kind of fictions, essentially. All right, so there's this kind of well-worn quote, right? All models are wrong, some are useful. You shouldn't ever trick yourself into thinking your model is real, right? Your model is a useful description of some aspect of the system that you're studying. Ideally, it extends quite far in terms of its utility and its explanatory power, but it's always going to be limited. Why do we do it? I think there's two main answers to this. One is that although in neuroscience in general, like the data we get really, it's always improving at a pretty impressive clip, more so in some fields than others. Data is getting better, but the data still is partial, right? We don't see everything that we want to see. We don't know the relationships between things that we measure in the way that we want. So to some extent, data is sparse, noisy, and incomplete. And the theory is kind of the glue, right? Theory and modeling and principles of understanding physiology. These are how we can connect this kind of sparse collection of things that we can see, like the kind of blind men on the elephant type thing. That's one of, I think, the most compelling way of thinking about what you're trying to do with computational neuroscience. But then something that comes with that, which I think is also important, is a certain amount of clarity and explicitness. that comes with writing down your theory at in the form of equations. Sometimes you want vagueness when you're working with certain kinds of research questions, but more often than not, it's good to be clear, good to be explicit, and that's something that you get when you're doing computational modeling. Okay, so I want to go do a very quick run through these three areas that I think it is worth thinking about how people approach and study stuff in computational neuroscience. So dynamics, cognition, and control. Okay. So when we're thinking about modeling and studying neural dynamics, the first thing that comes to mind is, and this is really important to understand, is just the range of spatial and also temporal. We had another axis there, the temporal scales that we have in brain systems, in neural systems. So this is one way of representing it. This is a slightly jazzier version of the same diagram. So there's stuff going on down at the level of microns, which will be pieces of a cell, up to the level of millimeters. That's now kind of, say, groups of cells, neural populations. Moving on up, then we're talking about things like patches of tissue, brain regions, parcels, Broca's area, things like that. And then all the way up to what we call large scale and even whole brain network structure. So these are all spatial scales at which people work at, people, put their whole career into trying to understand something at one of these levels, but there's always things above and below you, broadly speaking. And that's one of the to be honest, it's one of the exciting and fascinating things about neuroscience is this kind of multi-scale aspect to the way that neural systems operate. But that means that we have to, we have different structures at these different spatial scales, and we have different things to describe. Sometimes they have similar behavior. So there's something like properties of neurons that look a lot like what giant clumps of tissue do. Right? So kind of like general properties of excitable and oscillatory tissue that are shared. But there's a lot of differences as well. So you kind of need to know where you're at and what you're working with in terms of the structures that are relevant at different spatial scales when you're picking your computational models of choice. Two examples I'll show to kind of capture that. Some work at the microscale, very famous work from a project in Europe called the Blue Brain Project, which involved this kind of industrial scale reconstruction of, originally it was parts of the rat brain, rat somatosensory cortex, developed these physical geometric model of neurons for cortical columns, characterize their spiking rate, and have observed interesting things like emergence of slow periodic rhythms with calcium levels. These are very detailed, very computationally intensive models. They generate kind of petabytes of data per kind of second of simulated time. They're wild. So that's kind of very, if you like, granular models. And then a different approach, now more like at the top end of the spatial scales gap that arc that I was showing you before. So I'm just going to get my little video going here. And this is actually the kind of thing that we do in my group. Okay. I want to play something, but it's not. Okay, well, that's unfortunate. So I've got a little video here, but it's not playing, but this would kind of show you some of the things that are in some of these other slides. Oh, excuse me. Okay, so this is whole brain modeling. So this approach, what we do is we say, I do a MRI scan. I can reconstruct these things called fiber tracts, which in combination with some other information we can get from structural MRI about brain region locations and geometry. We can build these things called connectomes, which are large-scale whole brain connectivity matrices. And then we propose a relatively simple model of the activity of a large group of tissue, excuse me, say cortical column, and that gives us the activity for one of the nodes in our network. And this is like the way that we model and understand brain imaging data of the kind that we can kind of see from human subjects in vivo. So it's in an MRI scanner or an EEG or MEG, as I'm showing here. So the kind of things, for example, that we tend to study when we're looking at these macro scale brain activity patterns is a rhythmic behavior and rhythmic changes to rhythmic patterns as a function of disease. So, for example, the shift in your alpha frequency as a function of progressing dementia, changes in brain frequencies over sleep stages and over aging, and brain states. This is some work from my group from a few years ago. So this is a model of alpha activity, kind of 10 hertz rhythmic brain activity that we can that we see a kind of flip-flopping between this slow alpha rhythm limit cycle, so 10 hertz limit cycle, and a faster 40 hertz limit cycle. And this relates to kind of use of attention, engagement of attention in cognitive tasks versus a kind of idling resting state activity that we see with alpha rhythms at rest. So this is whole brain modeling. We also, some of the things we think about is what is the circuit structure for those nodes in the network? You know, should we think about them as corticothalamic loops, or should we think about them as certain types of schematic cortical microcircuits? And lots of fun things along those lines. So those are things that people thinking about when they're thinking about modeling neural dynamics. Few comments on cognition, coding, information processing. So David Marr is a very famous computational neuroscientist who died quite young in the 1980s. And he kind of laid out this proposal for how to think about how brains do implement information processing. Kind of three, they call them Mars levels. So there's they go in kind of progressive levels of abstraction or reverse levels of abstraction. So we have some computational theory about what is the goal, what is the strategy, what is the process. Then we have the representation. So this is kind of more an algorithmic design level, and then hardware. And in the kind of neurophysiology case, that will be kind of wetware. So how a neuron's implementing the algorithm. That's been quite an influential way of thinking about information processing in the brain. You know, that would be very much on the left-hand side of my schematic there. Other things that come up when we're thinking about cognition is learning. And in the neuroscience context, plasticity is kind of the implementation of learning effectively. Obviously this comes in, and there's a reflection of this in the modern AI and deep learning. This is which has come out of, to some extent, some of the understanding from neuroscience. But in terms of the structure of the brain, you know, there are certain different brain systems like the basal ganglia that implement certain types of learning algorithms. So the thinking is that basically basal ganglia is a reinforcement learning system. Cerebellum gives you supervised learning, algorithm implementation, and then the cortex, which is this kind of global computational general purpose tissue, does a lot of unstructured, unsupervised learning. So these are kind of ideas that are present. People are thinking about cognition from the point of view of plasticity. And one well-worked example of this, there's a lot of well-worked examples actually, but there's a lot of solid computational work about different forms, different types of plasticity, but the kind of mathematical structure of how learning is kind of implemented at the synaptic level in neurons. The kind of phrase that goes with that is basically like neurons that fire together, wire together, but there's a lot of interesting dynamics to do with like temporal coincidence and temporal kind of symmetries in how plasticity forms. So levels and algorithms and architectures, these are things that people think about in terms of cognition, but also some granular level of the physiology of plasticity as well. Okay, final point on my whistle-stop tour for this first piece. And this is one of the things that we also think about in my group is control. We broadly describe this as control. So this is often related to technology and therapies and interfacing with various kinds of, say, neuromodulation devices, but also drawing on principles of understanding control from other types of, say, physical systems. And one way of just describing that is just understanding the physiology of brain stimulation, which, as I mentioned earlier, is one of the things that we think about in my group. So the elements of that from the point of view of a computational neuroscientist are, there's a physics piece, right? So if we're doing TMS, as Parza was saying, he spent his master's working on, so we have some magnetic field that passes through the skull, generates secondary electrical currents in tissue, activates neurons, right? This is a biophysics problem. So there's a lot of kind of interesting problems that relate specifically to the kind of physics of electromagnetism in this case, or other types of physics for different types of neuromodulatory systems. And we'll do things like use mesh models of brain tissue from MRIs to solve these kind of Maxwell equations. A lot of interesting, just straight up biophysics that comes in when we're understanding brain stimulation. Then the next step in the process is, say, we've activated some tissue with magnetic fields or with electricity or with acoustic modulation, say with focused ultrasound. Great, what happens next? So this is now a kind of network question, right? So I've activated some tissue, like this red blob here. How does that propagate through to the rest of the brain and influence areas that are downstream? So this is a networks question. So there's an element of like reconstructing the networks, understanding their spatial structure, but also understanding the dynamics. So how activity flows through these systems. And then plasticity. So I mentioned plasticity before, that's a really important part of the therapeutic element of brain stimulation, because usually what we want to do is apply some, say, rhythmic stimulation that has a long-term therapeutic effect. You come into the clinic for a week and you go home and your depression is improved for at least a few months, right? So that's ultimately has to be a plasticity phenomenon. So plasticity is a really important element of the control question when we're thinking about brain stimulation. And then another, critical part of it, certainly in the therapeutic cases, well, how can you use what you know here, right? Like, let's say we understand a lot about how to induce plasticity. Well, then we need a kind of pragmatic and scalable clinical implementation. And that's another set of interesting questions that come once you've got your basic understanding. Okay. Sorry about the voice. I'm gonna do my best. Okay. So I was talking about propagation. That kind of second point in the diagram, and this is also the kind of thing Paz has been working on. just to give you an example, kind of visual example of this. So this is an evoked response from TMS. So these are EEG channel measurements in response to stimulation of the motor cortex. This is source estimated brain activity. You see kind of over time, following these arrows, kind of propagating out from the motor cortex through to, say, you see the blue bits now, frontal lobe, other hemisphere, out to the rest of the brain. So this is data. The bottom side is the model. It's actually what model that we've developed to try and understand these things. So one of the questions that we're often trying to characterize and explore is, how do we understand this propagation pattern? And we develop network models that try and replicate data in order to tease apart some of the features of it. Okay. And okay, final point, I mentioned plasticity. So just to kind of give you a specific example of this. So when we use TMS in the clinic, so this diagram here is a TMS coil. So this is your magnetic, a copper coil essentially that you run a current through, put it on the head, stimulates the brain. We're usually doing this with some kind of rhythmic pattern. So rhythmic pattern look a bit like this. We'll have, it's called intermittent theta burst stimulation, where we have bursts of usually 50 hertz pulses, 50 hertz bursts that are running on a carrier wave, which is usually about 5 hertz, hence the name theta burst. And then we have this intermittency, where we have this nested gamma theta stimulation train, on for 2 seconds, off for 8 seconds, on for 2 seconds, off for 8 seconds. So there's a lot of temporal structure in that. And the reason why that's kind of important is because the mechanisms in the brain that respond to rhythmic stimulation, they respond differently to different frequencies. And for example, one of the things that drives whether that rhythmic stimulation is going to push synaptic weights up or push synaptic weights down, so induce what we call long-term potentiation or long-term depression, is how it interacts with intracellular calcium concentrations. And this is very dependent on the frequency structure of the stimulation. So this is some of the areas that we go in terms of thinking about the plasticity aspect of brain stimulation from the point of view of computational neuroscience. Do similar things in deep brain stimulation for Parkinson's. I won't go into the details of that here, but similar kind of different set of questions, but interesting set of questions that are now more about like what's going on in this set of basal ganglia nuclei, where you have a implanted electrode that's activating tissue electrically, and a lot of interesting computational neuroscience questions there relating to control. Okay, so that's the whistle-stop tour of computational neuroscience. I just wanted to show you a few things that are hopefully exciting, interesting, with some applications, some real-world context, to give you a flavor for where we go with some of the tools that we'll now talk about for the rest of this lecture. Any questions on anything that's come up so far? Well, I take a quick breather. Nope. Okay. So we move swiftly on. Okay. So I want to spend some time talking about how we model and understand the neurophysiology of neurons, and particularly what we call single neuron models. Now, this is a huge field. I can't do it justice in one lecture, but I'm going to give you some of the basics, and skip some of the basics as well. So you look at a neuroscience textbook, you'll often see something like this, right? Kind of cartoon model of a neuron. You've got a cell body. You've got some dendrites, which are kind of where the inputs come in, if you like. And you have this long extension, a process called an axon that is often myelinated. That's what these light blue things are here. These kind of segmented pieces of fatty insulating tissue. And the way the neuron works is that there's something called an action potential, but which we'll get to, but activity is generated at the cell body essentially as a function of its inputs, travels along the axon, reaches the terminal boutons, releases some, usually, I mean, in the case of chemical synapses, releases some neurotransmitter that's then received at the next neuron in the chain, right? So that's your kind of textbook description of your neuron. It's pretty good. I mean, one thing I think is worth noting is like, they really don't look like this. What they look more like is kind of, imagine the axon is like a piece of string that goes, say, to the other side of the street. You know, the axon is extremely long relative to the size of the cell body. So this is a pretty kind of skewed representation, but obviously you can't really show that on a page. But the actual structure of neurons in real tissue is a lot of variability, a lot of structure and a lot of variability in the structure. But they're different variations of kind of branching structures into and out of a central cell body somatic compartment. Okay, so there's a lot of variability in the morphology of neurons, even though they all follow more or less the same basic plan. And then when we think about the kind of next level up, which is the kind of circuit structure, then there's a whole lot more variability and complexity that add on there. So this is like a classic diagram from Cajal, right? Just showing neurons that he's identified in a kind of stained section from a piece of tissue. It's really revolutionary work at the time. More recently, and I'm hoping my videos are working this time because I've got a little, no? Okay. Is there gonna be a way of, well. Yeah. No. Okay. Well, this will be a little movie from Blue Brain, which is a kind of fancy, detailed simulation of essentially this, but kind of really kind of showing you how the structure of a densely connected cortical column relates to how activity propagates through these branching structures. So it's just a nice kind of fun video. No critical didactic role other than that. So all of this is to say there's a lot of structure in neurons, not even thinking about the circuits, just thinking about individual neurons. And then how we approach that from the modeling point of view and the mathematical neuroscience point of view, we've got options. We've got options in terms of progressive levels of abstraction and simplification. So I'm gonna talk a little bit about some elements of some of these, not all of them. But broadly speaking, we can aim to describe, a complex cell in terms of everything about it in a nutshell, but specifically every part of its membrane and all of the branches, every little kind of segment of the branching structure, and have some physical and physiological description of that in the form of a mathematical model. Or we can abstract this. We can say, I don't care about all of this spatial structure, but I'll say, let's say that it's got two pieces. It's got a cell body and it's got some dendrites, right? So we call this like a reduced compartment model versus a detailed compartment model. Or we could reduce it even further down and say, I'm not even going to I'm not even going to suppose there's two parts of this. I'm going to say there's just one. I'm going to say this entire neuron is going to be described by, at this point, this is now like a point process where there's no particular spatial structure, but there's some kind of general activity that is the important feature that I want to be summarizing. And then there's different levels of, say, physiological granularity we can take for single compartment models, an abstraction, and even abstract them away from things like current flow, which we'll get to in a little bit, and talk about just things like activation functions. Now we're really looking at things that look like artificial neural networks in the machine learning sense, and even further to things like rate models, where we now have just some description saying the probability of firing at a certain frequency, and not even thinking about the initiation of even a graded firing per se. So those are kind of levels of progressive abstraction that we can think about for single neuron models. The very first step, so if you like this bit here, right? The place where it all starts is the membrane. So we need to talk about membranes. So if you look at an axon in an electron microscopy cross section, this can be something like a few dozen nanometers top to bottom, this kind of image. So this is like looking down in the middle of an axon. Okay. This rim on the outside, so this is the cell membrane, and the two kind of lines of the membrane, the two black lines, this is we call the phospholipid bilayer. This is what forms membranes in cells. Is these little molecules that repulse each other and form this kind of double membrane structure. So this is what cell membranes look like. You know, these bilayer structures, and then sitting inside the membranes, we have a variety of interesting and curious proteins that do interesting stuff. And we usually call those things channels, or ion channels. And they literally sit on the membrane and span the membrane, and they're kind of quite complicated, like chains of proteins that change their conformation depending on the electrical state of their inside and their outside. So what the kind of important thing that the membrane does from the point of view of neural dynamics and more or less everything that's interesting about nervous systems is that they generate this potential difference between the inside and the outside of the cell. So they polarize the cell, and they do this using this thing called a sodium-potassium pump, where they kind of actively push charged ions out of the cell, like across the membrane, so that we basically have a bunch of positive ions outside of the cell. So the blue is now outside, dark blue, light blue is inside. Bunch of positively charged ions outside the cell, and we have some negatively charged ions inside the cell. So we have two things now. We have a potential difference between the inside and the outside of the cells, more negative on the inside than the outside. And we also have concentration gradients for these specific species of ions. And there's several, but the main ones that really matter are sodium and potassium. So that's a thing that cells do, neurons do, other things do, muscle cells do it as well. They maintain this potential difference and concentration gradient difference between the inside and the outside of the cell, and that's what a polarized membrane is. Now, there's two from the point of view of the modeling and the dynamics. There's two properties of these charged membranes that we care about. And we call these the active properties and the passive properties. So the passive properties are like well described by a kind of simple electrical engineering concept, a resistive capacitive circuit. So we have inside the cell, outside the cell, is this circuit that has a resistive element and a capacitive element. The capacitive element allows it to store and maintain charge and kind of slowly discharge current, but not release all of its current immediately. And the resistance provides resistance to current flowing across the membrane. So this passive linear properties of the cell membrane, you can think about in terms of their response to an input current. So this little step function here, this is, say we're injecting an input current into the cell, and then we have a kind of blip and then a drop in the capacitive current, gradual rise in our resistive current, and the relationship between these two gives us the time constant for the circuit, and this is also is determined by the point at which the membrane potential reaches a certain proportion of its maximum. So this is essentially things kind of ramping up and ramping down in response to a stepwise current injection. Now that matters for the kind of dispersion of current both into the cell, like at a point. We think about one point in the membrane, we've got some current injection, but also laterally, right? So it really is kind of like water or fluid, like diffusing along the inside of the cell. So we have current spread along the internal part, the cytoplasm, and that's following these kind of passive RC circuit properties of the membrane. And there's a branch of computational neuroscience that I won't go into too much of detail here, called cable theory, that kind of has ways of finding correspondences between essentially idealized or simplified branching structures and more refined and kind of complex branching structures. But this is all still passive current flow through the membrane. So that's the passive linear aspect of membrane properties. So I had a slide title saying the neural membrane is an RC circuit. It's not just an RC circuit. It also has this nonlinear behavior, and that's the interesting thing, really. So the active nonlinear part of the behavior of membranes, neuronal membranes, is something that's determined by those sodium and potassium ions that we saw earlier. So what that looks like is, so we have the membrane potential here. The resting potential, which I didn't say earlier, so resting potential for neurons is roughly minus 60 millivolts. Okay, so essentially like neurons will sit with a potential difference between the inside and the outside of the cell of roughly minus 60 millivolts, kind of at rest. Now, what's going on here, so these little blips here, this is what I was showing in the previous diagrams as well. So now we're doing a little current injection, stronger current injection. So this is a current injection, and this is what the membrane potential response looks like. And this, so these two pieces here, this is an example of the linear kind of RC circuit component of the membrane dynamics. So when you give it a small nudge, you get a small nudge, you get a kind of quick response and then a slow decay back down, what we've got here. Okay. However, If you give it a big nudge, then we have threshold behavior. Okay, we have spike initiation, generation of an action potential. So an action potential, spike, synonyms. So if you push it hard enough, then we'll generate an action potential. So question is, how does that work? And that was the question that was, of interest to a lot of people in the what's sometimes called like the golden era of neurophysiology, which is the 30s, the 40s, the 50s of the previous century. But they're again really culminated in this work from Hodgkin and Huxley in the 40s and the 50s, got them the Nobel Prize in the 1960s, who did, basically the most seminal piece of computational neuroscience work in the history of the field. And that was in order to understand the physiology of action potential generation. So they were studying this thing called the giant axon of the Humboldt squid, I think. So very long, very kind of experimentally tractable giant cells. They didn't have very good gear in the 40s and the 50s, so they needed very big things. That's not really a problem these days. But what Hodgkin and Huxley proposed in their model of a generation of spikes is a relationship between the dynamics of opening and closing of two types of ion channels, sodium and potassium channels. We saw that the sodium and the potassium ions are moved out of the cell as it's by the sodium potassium pump in generating the resting potential. So the idea is, again, think about a current injection. So we've got a current injection. These are just the phases now. So the first phase, we're kind of ramping up. This dotted line here is our threshold. So when we reach our firing threshold, then our sodium channels open, and then we get a rapid influx of sodium ions. Okay, rapid influx, rapid opening of ion channels, followed by a rapid closing of the sodium channels, and then on a more slow, on a slower kind of time constant, when our membrane potential is in the positive domain, then our potassium channels open. They start opening here. So when you kind of cross into the positive membrane potential, but they're slower than the sodium channels. So they have this kind of more, hump-shaped response. So potassium channels open. We get our potassium flooding into the cell that then basically repolarizes the cell. We get this overshoot and eventually back to the resting potential. So it's this kind of flip-flop between opening and closing of sodium potassium channels that generates the action potential. Now, how does that look from the circuit point of view? So we had our kind of idealized RC circuit sketch that I had before. This is now the same thing, adding in a few extra components. There's actually more components that I need here. The main two are these two, the sodium and the potassium. But there are other ions that are kind of at play, but we kind of ignore them for the first level description of the Hodgkin-Huxley. So that's calcium and chloride ions, but the main things are sodium and potassium. So we have currents associated with the influx of the influx and the efflux of these ion species. And what that looks like is essentially the outgoing current of the cell is just the sum of the currents associated with each of these ion species. So that's the kind of key thing to understand about the Hodgkin-Huxley model essentially is is the kind of the sum of these currents that's generating the total current flux across the membrane. Now, the way that that works is related to these time-dependent conductances. So this is the ionic current. I is a function of this quantity GP, which is a function of the difference from the, folks on the call, still with us, okay. A function of the difference in the membrane potential to what's called its equilibrium potential. You think about that as just the potential that ion species wants to be at. So when that's large, this pressure, when this difference is large, this kind of current pressure is high. And when it's close to zero, when there's not much going on, this is low. So we have a current that's a function of this relationship. And the kind of key thing from the dynamics point of view is that this G here, this channel conductance is time varying. It varies over time, and it varies over time. It varies over time as a function of the voltage, but also as a function of the kinetics of the opening and closing rates of the ion channels. So the way to think about this, if I want to stack these together, I'm just going to get to this bit now. Here, okay. So putting all these together in the Hodgkin-Huxley model is a four-dimensional system of differential equations where we have one the primary equation, if you like, the first one. So this is the rate of change of the membrane potential is a function of the four input currents. Sorry, the two ion channel currents that I talked about. This thing called a leak current. That's the passive component. So that's like the original passive membrane property I talked about before, and the injected current. So usually we're thinking about how the neuron responds when you insert some current to kind of push it over the threshold. This is the first equation. And then what we have kind of embedded in this now is our next three equations. And these are the time varying behavior of the gating variables for the respective ion channels. Okay, so we've got a term called we call it gating variable. Each of these things here, which has it's essentially, it describes the passage, the movement of a concentration of one substance to another as a function of a rise time and a decay time. So we have two channel probability terms that are related to the sodium channel, because the sodium channel has rapid opening. You remember the figure that I showed you before? So we have essentially our sodium conductance goes up quickly and it goes down quickly. So the reason why it goes up quickly is we have an opening probability. We have gating variables related to channel opening, and then gating variables related to channel closing. The potassium current, on the other hand, we don't have a rapid closing. We just have a slow opening and then decay. So we have two gating variables associated to the sodium conductance. Come back to here now, which is our M, our N, and our M.
And then we have just one gating variable related to our potassium conductance. And essentially these guys are both fast and this guy is slow. So when we put that together, put all of these together, so if we put, say, like this, plus these three, gives us the full Hodgkin-Huxley model. It's a four-dimensional system of nonlinear differential equations, membrane potential, and then our three channel. Wow, I keep dropping the term that I'm looking for here. The channel, the gating variable, opening dynamics for our 2 sodium and our 1 potassium gating variables. Okay, so when we kind of look at that in action, this is quite a complex figure, so bear with me. This is the entire process, all of the things we've just talked about all together. Okay, so this is the top row here is the membrane potential. So we have, and the membrane potential in response to a current injection like we saw before. So the first one here is a small current injection, as we saw. So we're getting we're not crossing the threshold. So we have this passive current response property first. Nothing much going on essentially. And then our second example here, we've got a larger current injection. This pushes us over the threshold. Generation of an action potential. The action potential is defined essentially by this transient switch in the membrane polarization, goes positive, then goes negative again. And we see our gating variables tracking this now. So our sodium and our potassium gating variable, H and N, are rapid, the opening and the closing variable are, so our, N and M are the 2 sodium ones. So M kicks up super quick and then drops down. And then conversely, H drops down quickly in response. And then our slow thing is the potassium one is N. So this is kind of ramping up slowly and then rolling down slowly there as well. So the gating variables, those are our second three terms in our 4-dimensional differential equation. And then these kind of group together into the conductances, which is the kind of current terms respectively for the sodium and the potassium channels that are giving us the rapid depolarization followed by the slow repolarization in the membrane. Okay, so that's Hodgkin-Huxley. You could do like 3 lectures on Hodgkin-Huxley. So if that was it seemed like a lot, it was a lot. It was a lot with a lot missed out, but I tried to give you the kind of essentials. Any questions? Any questions on the chat? No? Okay.
Yeah, we do have a question. Hi, Professor John. So if I understand correctly, we must find a solution for the membrane potential V, which will be substituted to the 3D system governing the probability dynamics. Then solve the probabilities for some initial conditions. Also appreciate it if you can explain once more what the probabilities mean qualitatively.
Yeah, sure. Yeah, so we're solving the. I mean, you're right. We're not solving this system in terms of a fixed point or a steady state. This is like a numerical integration for a given set of starting conditions. I think that was the first question. What was the second question? What's the interpretation of the gating variables?
What is the qualitative interpretation of the probabilities?
The probabilities, yeah, probabilities, okay. So the probabilities, I skipped over this a little bit, is what's the best version of this here? Probably here. Okay. So I'll start just with the diagram to give you a starting point. So what we're talking about here is, qualitatively speaking, also just literally speaking, like this is the structure of the membrane proteins. They have three different, the sodium protein, sodium channel, has three different states: closed, open, and inactivated. And it basically moves between these states. Okay. Now there's a probability associated with each of these that is to do with the channel structure. It's kind of represented in this diagram a little bit. So there's 4 components to the channel structure for sodium. I think that's the right way around. 4 for sodium and 3 for potassium. We check this with the number in the equation that we'll get to. Bear with me. Yep. Okay. So 4 for sodium. Sorry, 3 for sodium, 4 for potassium. So the probabilities are to do with the state of the ion channel, and whether it's in the case of sodium, whether it's in open, closed, or an inactivated state, and then moves between, it's cycling between those 3. The reason why we have exponents of n to the 3, n to the 4 here is to do with the number of subunits in the channel protein. And essentially the protein will be open when all of those subunits are open. So it's, a probability or a term raised to the power 3, because we need 3 things to be open at the same time. So we multiply the probabilities in order for something to flow through. So that's kind of where we get these exponents in the probabilities in the gating variable terms. And yeah, I mean, qualitatively, it's the rate at which the ions are flowing through the channels. That's the best way to think about it. The reason why we've got, yeah, I think I'll leave it at that for now, but great question. And let me know if there's anything further you want me to clarify. Okay. Any other questions? All right. So let's keep going. So we talked about Hodgkin-Huxley there from the point of view of the kind of biophysics, and the ion channel properties and the ion channel physiology. We can also look at this from the perspective of a more abstract dynamical systems point of view. So the process, I've seen this diagram a few times now. So we've got current injection, whether it's slow or fast, we have a period where we have current ramps up, crosses the threshold, nonlinear behavior here, recovery, go back to baseline. Okay, that's the qualitative dynamics here, right? So the existence of the threshold here, call it threshold. It's an all or non-event, right? And it occurs once the membrane potential exceeds some threshold. So at a simple level, and now we're kind of moving to some visual representations of dynamical systems. So you can think about this. If you think about this as just a simple, like one-dimensional dynamical system, this is kind of a sketch. Okay, so so this is a standard way of representing graphically, visually, the like dynamical structure of a phase portrait of a dynamical system in one state variable. The state variable is V, and it's the value of the state variable is the location on this horizontal line. Okay, the got a visual language for this, these kind of graphics is, so we have our axes here. So in this case, as I said, V is the value of the state variable. These dots here, these are intersections. Excuse me 1 second. So these are points when the state variable is 0. Okay, so this is 0. This is crossing here. This is the amplitude. Why am I? Yeah, dV. Okay, so the y-axis here is when we're talking about a one-dimensional dynamical system is the derivative of the term that's on the x-axis here. So y is v dot. Okay, so when v dot is high, then, or when this line is here, we're saying the kind of rate of change is high, and then as we go down to here, so this value, the rate of change is 0. Okay, so these circles on our horizontal line here are points at which the rate of change is 0, and those are what we call the fixed points of the system. Okay, so when you're on a fixed point, you're not moving, you're going to stay still, you're at a steady state. Now there's two, as you can see, kind of types of circle here. There's circles that are filled and circles that are empty or kind of black in the middle. And that this is the kind of standard visual language for an attractive versus a repulsive fixed point. So essentially the phase flow, which is another way of saying the movement of the activity variable, up and down. So V goes up or V goes down. V goes up, be going to the right, V goes down, going to the left, is moving either towards one of these fixed points or away from one of these fixed points. And this is a kind of simple structure for a one-dimensional dynamical system. We have 3 fixed points here. The one in the middle is unstable, it's repulsive. Okay, so if you're anywhere other than directly on top of this value of V, you're going to move usually rapidly in this direction away, or you're going to move rapidly in this direction, and most likely you'll end up at this stable fixed point and you'll stay there. Okay, so that's kind of for present purposes, this is a schematic of thinking about the qualitative dynamics of a threshold behavior. Okay, because the point of a threshold, go look at the previous slide, right, is you have this, say, this passive current response. Either you cross the activation threshold and you have one type of thing, or you don't cross the activation threshold, in which case it will decay back down to its resting state. Okay, so like A or B behavior. So that's kind of what this is basically showing. Like if this will be decaying back down, not crossing the spike threshold. Or if you have enough of a current injection, then you cross the spike threshold, I'll move you to some other state. Okay. So this is one dimension. This isn't really what Hodgkin-Huxley models look like, neural dynamics look like, but it's a starting point. So there's a number of simplifications of the system we were looking at before, Hodgkin-Huxley system. Oh, sorry. Yep.
Just out of curiosity, is this threshold sort of uniform in all parts of the brain, or like systematic?
I think it's pretty consistent. I mean, I'm sure it's not completely identical, but I'm not aware of it varying greatly. I think maybe like plus minus 10 to 20 millivolts, but it doesn't vary hugely. But I mean, another thing that's relevant there is that you have the property of, say, a membrane, a kind of idealized membrane, but then you have the naturalistic state of the cell. And the naturalistic state of the cell is pretty complex and dynamic. It's going to have like some inputs at a certain time, in which case, you know, its threshold is wherever it is, but it's going to be close to the threshold because it's already activated, right? So the kind of behavior of neuron is very dependent on context, whether the threshold is, you know, moving around or not. Okay, so if you recall, the kind of full Hodgkin-Huxley system that we talked about was 4-dimensional. We had the membrane potential, then we had our 3 gating variable terms. And the key thing about the gating variables was the sodium one's fast and the potassium one is slow. Now, there are a number of models and approaches that have been developed over the years, the 80 years since the original development of the Hodgkin-Huxley model, that simplify that system down to a more kind of conceptually and mathematically tractable form. There's 2 that we're going to talk about today. One's called the Morris-Lecar model. The other one in a few slides called the FitzHugh-Nagumo model. Both of these are 2-dimensional reductions of the original 4-dimensional Hodgkin-Huxley equation. So the way that the Morris-Lecar model achieves this is by basically dropping the sodium conductance. And the way it drops it is by assuming that it's instantaneous. So those 2 terms, those 2 gating variable terms in the 4 differential equations are basically dropped. And then all we have now is 1 equation for the membrane potential and 1 for the gating variable for the potassium, which is this n variable here that we've got a second equation for here, okay? So we've dropped sodium, we've just kept potassium. What that boils down to is we've got a fast variable and a slow variable. And that is basically what the essential dynamical structure of this system is, and of a lot of systems are. Kind of general purpose technique in terms of analyzing and trying to understand systems is timescale separation. So we found a fast piece and a slow piece and kind of ignored some of the finer grained details. I'll explain a lot more about that as we go. But the second point that we've got here on this slide is talking about the null clines, right? So when we have particularly a 2-dimensional system like this, well, also the 1-dimensional system, we can study these phenomena called null clines. These are the 0 points. So these are the points for which the rate of change of the state variables is 0. So depending on the structure of the equations, the null clines are going to have a certain shape. But what they look like visually is the following. So we have now our 2-dimensional Morris-Lecar system. We have the membrane voltage equation. And as I said, just one of our original 3 gating variables, the slow potassium one. So we have a 2-dimensional system, and we can represent the entire qualitative dynamical structure of this system in a kind of square block of graphical information. So what we've got going on here is, so where any point on the plane is a value of each of the 2 variables, okay? The background here is what we call the phase flow. So these arrows here, these are telling us for a given point that you are, what direction are you moving in? Okay, so if I'm here, then I'm moving, up and to the right. If I'm here, I'm moving down and to the left. What does that mean? That means the membrane potential is going up and the gating variable is going up. The membrane potential is going down and the gating variable is going down, right? That's what the directions in the phase space mean. That's also what they meant in the 1-dimensional case. But when in the 1-dimensional case, we had a another dimension there for the derivative. We don't do that when we're showing a two-dimensional system, but the kind of up and down in the one-dimensional case, that's basically the same as up and down in the two-dimensional case. We just also got another dimension to move in. Okay, so that's the background phase flow structure. And I mentioned the nullines. So the nullines are these lines here. Okay, we've got two nullines because you have two variables. So what they tell you is at each point, at any point on the nullline, excuse me. So this one is the nullline for V. Yeah, it says there actually. As you can see, it has a cubic structure, so it goes down, up, down. And the nullline for K is quadratic, so it kind of just kicks up. So at any point on this thick line, the rate of change of V is 0. That's what it means. Okay, and same here, rate of change of N is 0. So then the intersection of the nullines is the definition of fixed points. So that's when the rate of change of both of the state variables of the system is 0, and you're at a fixed point. The activity is not changing, it's staying still. Okay. And as we had before in the one-dimensional diagram, we've got a solid fixed point here. So this is telling us this is an attractive fixed point, a stable fixed point. So the phase flow is moving towards this point, and when it gets there, it's going to stay put. Okay. So we're gonna track the evolution, the modification of this phase portrait as a function of one of the variables of the system, one of the parameters of the system. And that parameter is the input current. So going back here, is our input current over here at the front. So we're gonna increase input current and see how this phase portrait changes. Okay. So this is for low input current, low I. And as I said before, we have fixed point here. We're gonna kind of see how this changes. So this is kind of what you want to be looking at. So low I. Now these lines here, these are now trajectories. So this is when we've tracked the activity of the system for some starting point. And what it's done in all of these cases is it's followed a little loop where one variable's gone up, the other one's gone down, and then it's looped and come back around to the fixed point, right? So it's kind of reached this stable point, and then it's going to stay still there. Okay, so these are trajectories in our phase plane. There's like four different trajectories of five different initial conditions. The difference in the initial conditions being the value of the membrane voltage. And you can see when we start with a larger membrane voltage, we have a larger excursion, and we actually have this kind of threshold behavior. So for a low level of the membrane voltage, a low initial condition, we have a subthreshold response. For a larger initial condition, we have a large looping transient, and this is what we call what an action potential looks like in the phase plane. So this is the structure for low level of input current. Now the thing we want to track now is how that changes as the input current goes up. So this is now a little bit of an increase in the input current. You can see this, basically these small transient, the kind of damped oscillations are getting bigger for the same input levels, and we have more of these lines that are moving onto this large action potential manifold for larger levels of I, but we still have the same basic structure. We have our fixed point here, and we have we have large but damped oscillations. So it's still coming back to zero. Now we've reached for a larger level of our bifurcation parameter I, a qualitative change in the system dynamics, which is called the bifurcation. Specifically, it's a subcritical Andronov-Hopf bifurcation. So what's happened here? Again, it's all defined in the visual language of this two-dimensional dynamical system. So we have our stable fixed point has become unstable. And unstable, you represent that with an empty hole, an empty circle. And the qualitative change with the high level of the input current is the transition to what we call limit cycle. So now we have any starting point in the phase plane will move onto the manifold, onto this circle that defines the limit cycle. That is now a repetitive, sustained, oscillatory, recurrent inflection. So it's going round and round and round and round and round, right? It's not going back, excuse me, it's not going back to zero like it is here. Here it's going round, but once it's gone round, it's gonna go back to the fixed point. As we move through the Hopf bifurcation now, it's going to go round and then it's going to keep going round, keep going round, keep going round. And what that means, going round and round and round and round, is this. There's an oscillation. And up and down and up and down and up and down, as opposed to up, down, and then maybe back to a flat line, stable point after a damped oscillation evoked response. So that's one of the key things that we can study and is of interest from the dynamical systems perspective on systems like simplifications of Hodgkin-Huxley, like the Morris-Lecar that we just looked at. This is just summarizing what we showed then kind of all together, so you can see it, I guess, a little bit more clearly, right? So increasing I, we get larger excursions, then we get evolution, then we get, you can see that actually a kind of spiral here. So you see it spiraling back, spiraling down into the fixed point here. And then here's the transition, right? The transition here initially is a kind of low amplitude limit cycle. We crank up the current, we get a higher amplitude limit cycle. And that's all as a function of this bifurcation, what we call the bifurcation parameter, which is the input current. Okay, so that was a Hopf bifurcation. There are other interesting qualitative features that we can see in this system. I'll talk about one more of these, and that is the existence of the threshold, which we also saw in the one-dimensional case. So this is so the phase portrait here, you can see this time we've got three fixed points, so three circles. The circles are always the points where there's an intersection of the null lines. Okay, so the thing that determines whether there's 3 or 2 or 0 or 1 fixed point is just literally like whether this line is going to come down and up and, you know, intersect this one twice, or is it just going to kind of pass through it once, right? That's actually what, like, geometrically determines the number of fixed points. So in this case we have 3, and we have 1 that's unstable, sorry, 2 that are unstable and 1 that's stable. Now this one over here, this is called a saddle node. Sorry, I should say this is called a saddle. I guess right. This is called a node, and this is called a saddle. And the emergence of the the destruction of the saddle and the node is called a saddle node bifurcation. So I don't have a diagram showing the kind of progression of it. But essentially what happens is as we move through the bifurcation parameters, so I think we inject current, same way we did before, these two fixed points literally collide. They move towards each other up until a point when they're on top of each other, and then they disappear. So that's kind of geometrically what happens if you like track the evolution and we'll look at the phase plane. And what that corresponds to is the existence of certain states, and then the loss of those states as kind of available regimes in the system. So in this case, what we're talking about here is this resting state. So it's kind of the difference between the presence of a stable resting state or the sole existence of the limit cycle, okay? So in the previous case, say here, so here, the only thing that you can do is be on the limit cycle. Like anywhere in the phase plane, you're gonna move, you're gonna follow the lines, and you're gonna end up on the limit cycle. So there's only one dynamical regime that's available in this configuration. In this configuration, there's more. Okay, so you could have you could have excursions around this fixed point, excursions around this fixed point. You can move towards this fixed point. So there's a few things that could happen. And one of the terms that kind of describes this presence of a regime over here and a regime over here, where if you're on this side of the gap, it's this structure we call a separatrix. So separatrix is a manifold through the phase plane where the phase flows separate, such that if you're on this side, then you'll move in this direction. On this side, then you'll move in this direction, and you'll go one way or another. And that is essentially your threshold behavior that we saw earlier on. So the saddle node bifurcation is where we have the separatrix here. So you're on this side, then you move to rest. If you're on this side, you'll move on to the limit cycle. The saddle node bifurcation is the destruction of the stable resting focus here, leaving only the limit cycle. And then we can see that here actually qualitatively. So, well, actually, no, this is just showing the bistability, right? So, if you're at rest, then you're going to stay at rest. If you're spiking, you're going to stay spiking, and you're not going to move between the two. So the separatrix in the phase plane here, this line here, this is basically like this level of the amplitude of the membrane potential, which is corresponds to the threshold, the firing threshold. Okay. I think that's everything on dynamical systems. Any questions on the last few bits? Nothing burning. Okay. So I'm gonna move fairly quickly through the last few sections, and then we're gonna wrap up the first part of the session today. Then we're going to take a break. We'll take a 10-minute break, and then we'll do the hands-on tutorial demo to wrap up in the final hour. Okay, so let's talk a bit about firing rates. Now, we looked at a few examples of where we inject a current, and then we kind of move a neuron from a resting state into a firing regime. So we've got action potentials. This would be a limit cycle here. Okay, we've got repetitive firing for a given amount of an input current, stable oscillatory activity. And one of the features of neurons and good models of neurons is that the more input current you inject, the more firing you get. So example here, so like higher level of input current. Here we don't even get no firing at all. This is just the passive response. Firing, faster firing, okay? So this is a general feature of how neurons behave. And the term of art for this is called an FI curve, or a frequency current curve. So the input, this is the input current on the x-axis, the firing rate, so the number of spikes per second. And then as you can see, so basically once you cross the threshold point, more input current you put in, the more spikes you get, right? So FI curves, they look something like this. Skip this for a second. So the way that we, I'm gonna kind of move into this will do. So the shape of the FI curve varies for different neurons. The shape and the kind of rate at which they respond. And this is one of the kind of most important physiological properties of cells. So in those examples of kind of complex multi-neuron, maybe even multi-compartment simulations of neural circuits where we have multiple cell types, well-defined cell types, this is one of the properties that you want your model to be capturing. And you can achieve it by just having different values of the parameters, some of the parameters that we talked about, some of the conductance parameters. But essentially, like different neurons should have different FI curves, and this is something that can be incorporated into extensions of the Hodgkin-Huxley type models that we've been looking at here. So this is a sketch. This is maybe a little bit out of sequence, but it's an important diagram. Talking about different types of neuron model. It's kind of pretty famous figure from this very famous book from Itzkevich. And the summary here is the kind of computational cost of a given model versus the biological plausibility. This is the kind of qualitative axis on the Y here. And these points are different types of neuron model. So we've talked about some of these already. We haven't talked about all of them. But obviously Hodgkin-Huxley is the first one we were looking at. So this is considered highly biologically plausible, but quite numerically intensive for most purposes. And from Itzkevich's point of view, that's not great. What Itzkevich wants to say is that what you want to be is underneath this curve, and lo and behold, the best neuron model that sits underneath the curve is the Itzkevich model. But that's kind of why people like the Itzkevich model. So I'm not going to show you Itzkevich model. We did look at Morris-Lecar, and I'll talk to you about FitzHugh-Nagumo and briefly integrate and fire. But we'll think about these two at least. So we had, if you recall, we had fairly similar behavior between the Morris-Lecar and the Hodgkin-Huxley. That was the point of having this, all the stuff with the phase portraits. Right? And the point of that is to show that the essential dynamical structure of that four-dimensional nonlinear system, the Hodgkin-Huxley, is captured well by the simpler two-dimensional system. So it does well on the biological plausibility with less numerical intensity. Similarly, FitzHugh-Nagumo, which we'll touch on in a bit. And then moving back over here, we have things that are less biologically plausible, but also more kind of numerically feasible. And in this regime, we're talking about models of neurons that typically don't have much to say about things like ion channels and ion channel conductances. So when we're kind of up around this part of the diagram, we're usually talking about models that are saying, spike or no spike, initiation of a spike, but not necessarily anything biologically accurate about the amount of sodium, for example. Okay, so there's one other simplified two-dimensional model. This is one of the ones on the diagram just now that I want to point to. I'm not going to say much about it other than this is essentially exactly the same as everything we were looking at with the Morris-Lecar. It's a two-dimensional reduction of the Hodgkin-Huxley equations called the FitzHugh-Nagumo equation. It was independently developed by Richard FitzHugh and Dr. Nagumo, and in the 1960s, so before the Morris-Lecar model, and it's kind of a little bit more biologic, a little bit more abstract in terms of the meaning of the state variables. So the Morris-Lecar model, we had this the first term was essentially the membrane voltage. The second term was the gating of the potassium conductances, potassium channels. In this case, we've got variables that are qualitatively similar, but we don't necessarily label with the kind of ion channel species. But the important thing is we've got a membrane potential-like variable, X, or sometimes it's V, and a fast, and importantly, it's a fast variable, and then a slow variable. And the slow variable is usually we call it recovery variable. But the structure of the FitzHugh-Nagumo really exactly the same as Morris-Lecar in the sense of we have these trajectories in our phase plane that move through bifurcations and describe a lot of the phenomenology of how neurons behave, such as generation of spikes at different frequencies, generation of damped oscillations, overdamped activity, etc. Okay, so one more model, and then we'll call it a day. There's many models. We could be talking about models until the cows come home. This is a very simplified single neuron model. It's called leaky, integrate, and fire model. And if you recall the diagram that I had before, this will be on the top left. The top left is in very numerically computationally efficient, but not very biophysically detailed or plausible. So the basic structure of the leaky, integrate, and fire model is we have it's a now one-dimensional system, so one-dimensional differential equation that has a kind of logical behavior on a threshold parameter. So we have this leak current behavior. This is basically exactly the same as the passive membrane properties in the RC circuit that we start with, or if you like, the leak currents in the Hodgkin-Huxley. So this is our kind of passive membrane properties. And then the kind of addition in the leaky, integrate, and fire is that if the membrane potential crosses a threshold, then reset. So essentially, we just say, if V crosses threshold potential -50, I think, millivolts, then we just set it to 0. So not set it to 0, set it back to the resting potential. So what that captures is two features of the behaviors of neurons that are kind of often the two that we care about, two that we talk mostly about, is this linear subthreshold behavior, and then the nonlinear or non-behavior with the spike. But a very, very simple equation, one-dimensional, doesn't have a lot of elegant dynamics per se. It's kind of unstable, or singularities around this threshold, so isn't very kind of mathematically well behaved in the area of the threshold point, but is a very good workhorse for simulating neurons, which is often basically what we want to be doing. For example, this is a limit cycle, effectively, or the analog of a limit cycle with the LIF model, with no noise and with noise. So this is another type of qualitative dynamics we can add in. So we add, so we have some current coming in. See, the current is pushing the state variable up. So it's threshold point with this capacitive leak current kind of slope. It reaches it, then we reset it back to its resting potential. And then it drifts up again, reaches the threshold, reset, 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 right? That's no noise. Add in some noise, we get obviously like a bit of a variation on this, but this is actually, you know, how neurons look in the real world. Like, they're noisy, they have randomness and stochasticity. So just adding a little bit of noise on top of this simple system generates a kind of irregular spiking behavior that still will show things like Fi curve properties with input currents and some kind of lawful behavior, but a certain level of randomness that is, you know, physiologically realistic with a relatively simple model. Okay, so I'm going to call it a day. We had one more model that we could talk about, but I'm going to skip, which is the Poisson model. I'm going to finish with a brief comment on multi-compartment models, and then we'll take a break. So multi-compartment models, and this is just a passing comment, essentially, to relate to one of the first points that I made. So I talked about progressive levels of simplification, right? So this would be, say, lots of small pieces, right? So broken down into a lot of spatial detail. This would be a detailed, what we call multi-compartment model, where there's lots of patches of membrane there. Each of those patches of membrane would be the way that these models are usually constructed, following Hodgkin-Huxley equations, and actually extensions of Hodgkin-Huxley equations that have things like calcium and chloride and magnesium and other exotic ion species. And then progressive levels of simplification. Simplification down to this thing, which we often kind of affectionately call like a spherical cow. Okay, so these are different levels of simplification of multi-compartment models. The general point here is essentially, like, depending on what it is that you're trying to describe, often it's now something about the geometry of a circuit when we're talking about having these kind of, you know, 1, 2, 3, 5, 10 branching structures. Then you'll describe your neuron with these, you know, blocks, and then each of those blocks is going to be described by those usually Hodgkin-Huxley type ion channel behavior, plus maybe some of the passive cable properties of the current flowing through the inside of the cell. So these are called multi-compartment models. Okay, so here we go. That's a wrap. We had basically 2 lectures in one here: intro to whirlwind tour of a few interesting things in computational neuroscience from my point of view, and then a reasonable deep dive on some of the nuts and bolts of single neuron models. I want to remind you, you might to think of these 2 terms as synonymous. We have multiple spatial and temporal scales in neural systems. We've really only, at least the second half of the lecture, we talked about one spatial scale, but maybe some levels of progress, a few different levels of simplification at spatial scale, i.e. like the level of a single cell. What we'll be covering in the subsequent lectures is moving up the spatial scales, broadly speaking. So we'll think about networks and kind of statistical properties of neural ensembles, often constructed from the single neuron elements we talked about today. But yeah, there's a lot of spatial scales to span. I didn't say this, but okay, I should probably tell you this. So the terms that you see in the literature are usually these three, okay, micro, meso, and macro. The problem here is that they're not really well defined. Well, the only one that's really well defined here is micro, in the sense that it basically pertains to microns, roughly. But the thing that you'll often find, and it is sometimes confusing, is like one person's micro is another person's meso, one person's macro is another person's meso, etc. You know, it's really like people who work on, say, LFPs in DBS think of EEG as macro and, you know, single cells as micro. People who work on EEG and fMRI might think of LFPs as fairly micro, and so on and so forth. So, you know, expect some variability in use of these terms, but, you know, they go from small to large at least. I gave you some ideas, some suggestions to think about dynamics, cognition, and control as elements of how people think about computational neuroscience. And then we did a reasonable, as I said, deep dive on passive and active membrane properties, Hodgkin-Huxley, Morris-Lecar, actually more than FitzHugh-Nagumo. But the reason why I mentioned FitzHugh-Nagumo mainly is because that's what we're going to look at in our hands-on session after the break. So without further ado, any questions on anything that I've said in the last 2 hours? Now's your time.
Professor? Hello.
Oh,
Yeah, am I audible?
yeah, go ahead.
Professor, so there was that one-dimensional bifurcation diagram that you had for the membrane potential, right? Is it
Yeah.
possible to go back to that slide?
Sure.
Yeah.
Yeah. Yep.
Yeah, that one. Yeah. So this seems a lot like a supercritical pitchfork bifurcation, right? If so, like, what is the parameter that's varied to achieve this behavior?
Well, there's no parameter being varied here because, oh, you mean the, okay, you're talking about this, these
Yeah.
lines here.
The fixed points, actually. So there's two stable fixed points and there's one unstable fixed point.
Yeah.
So does it like switch from stable to unstable and it gives birth to two new fixed points, or is it something different?
Yeah, I mean the use of this figure was mainly intended as a heuristic. I wasn't actually trying to make a point about the bifurcation. I just wanted to talk about like a one-dimensional phase flow.
Right.
And it's not actually, basically, I don't know is the answer to your question. Like this figure is, sure, I don't remember what F is in this equation. But the point of the diagram was to just demonstrate what a one-dimensional phase plane looks like with some stable and unstable fixed points. Is that satisfactory?
Yeah, that's fine.
Yeah.
Thank you.
Okay.
I think the closest thing, I mean, we're not seeing a bifurcation yet, but I think the closest bifurcation I can think of is a Saddle-on bifurcation. Where you, for example, when you're seeing the null line, you just shift it up a bit in your mind, you can see that the stable and the unstable fixed points on the left side would converge closer together. and at some point they would merge in a sense, and then they would annihilate each other. In
Yeah,
that case, you would only be left with one fixed point at the top right or the far right. And this would be more representative of a satellite bifurcation.
I think that's right, but I'm not 100% sure in the 1D case. I think what he was pointing out about indicating a bifurcation is these dotted lines, kind of showing the movement of the fixed points. But yeah, Paz is entirely right. You know, the movement of, you think about this cubic line, right, just shifting up. Eventually, there's just going to be one part of the line that's touching the horizontal line, like right in the middle here. There won't be two, so there'll be a collision of these two fixed points. And a similar thing, which I didn't exactly say, but while we're on the topic, is yeah, I want to, well, I was going to talk about the way in which the fixed point falls on the descending or the ascending limb of the null line. But yeah, maybe I won't go into that. That's a little bit of a long discussion. But yeah, great question. Who was it who asked that? Was it Ravindu?
Yeah, Ravindu.
Yeah, fantastic question. Thanks. You had another question with it?
Which model should
Yeah, that's a very good question. Which model to choose? Oh my God. Okay, let's find the Itzhak diagram and talk about this. So, I mean, the obvious, the boring answer to the question is, it depends what you want to know, right? Or it depends what you want to study. But the point of this diagram is to say at least one way of answering the question is it's often a bit of a trade-off, right? So it's a trade-off between detail and feasibility, right? And feasibility, there's a few elements to it. One is numerical, but another is kind of conceptual, in the sense of one of the criticisms of, you know, the video that I couldn't pull up. I might be able to just cancel the slideshow and play it, this thing. See if okay, I think people on the call might not see this, but just they can. Okay, oh great. So the video that I want to play is this. Oh wow, there's music. Okay, didn't know there's music. Oh, but you can't see that. Let me try and share my screen on Zoom. Bear with me one second. This is all in the service of answering your question. Okay. Yeah. Okay. So it should be on the Zoom now, but no, we're not seeing it here. Okay. Do we need to move this onto the other? There we go. All right. So this is the video I wanted to play before. So this is a very detailed model of. Oh no. Okay. Well, I don't think. Okay, guys on the Zoom, I'm not sure I can show the people in the room and the people on the Zoom the thing at the same time. So I'm going to show the people in the room first, and then I'll put it on the Zoom, okay? So just bear with me, because, you know, why would life be simple? Okay. So this is a very detailed model of the brain, of a circuit. Oh no, come on. the point that I want to make here is there's two things that are challenging about very detailed models. One is that they're numerically intensive, as I mentioned before. So this is, you know, hundreds of thousands of cells that are being simulated with full morphological detail. So like every section of the membrane, right? And a lot of synapses and a lot of stuff going on. So this is, this is for like a piece of tissue that's a couple of millimeters max in size, okay? Generates petabytes of data for like a second of simulated time. So that's computationally difficult, right? It's computationally difficult to simulate. It's computationally difficult to analyze the results of the simulation. It's difficult to even just store the results of the simulation, right? And usually you don't store much. You simulate stuff and then get some abstractions. So there's lots of challenges for like numerical challenges with complex models, okay? Although the complex models are usually like our best, you know, granular description of reality, of biology, okay? But there's challenges numerically, practically, and then there's challenges kind of conceptually in the sense of, okay, well, I've generated petabytes of simulated neural activity. What do I do with it? Like, what's the understanding that I'm looking to gain from this complex model that is, you know, possibly almost as complex as the thing that it's trying to model, right? So there's a bit of there's pros and there's costs that come from the practicality, and there's costs that come from at the conceptual level of like what you're going to get your insight from if you have a very, very complex model of a very, very complex biological system, okay? Now, on the flip side, you know, considerations like why choose this model, why choose that model? That was the question here. I'm going to go back to the cage again. If I can find the mouse.
For example, in computer science, they shift from sigmoid to the loop, and they significantly drop the computational cost.
Yeah. Yeah. So right. I mean, it's exactly the right question. So I think the challenges come in all of the details, right? So let's take the LIF, right? So simple model is actually like versions of this are used a lot. Okay, so the problem with this is that it does weird stuff around the threshold. Okay, so it's kind of numerically unstable for fast stuff to do with fast spiking. It's okay for certain behaviors that neurons do, but if you want the full kind of dynamical repertoire, then it's not going to work. Okay. You know, likewise, so I was saying the structure here is essentially up here, you're usually saying, I want to know about firing, right? And over here, you're saying, I want to know about chemicals. I want to know about conductances and like physiologically measurable ion channel properties, right? So that's your kind of abstraction level going from the top left to the bottom right. Like talking more about things like wet biology things. Over here, more about kind of approaching binary digital type descriptions of like a neuron being on and off, okay, or on and off with a little bit of a recovery, you know, dynamics. So that, I think that's the other, maybe another part of answer to your question is like, do you care about chemical flux measurements that you can, see on a dish, or you can measure in a mouse? Or do you care about whether neurons are on or off? Okay, if you care about neurons are on or off, then you'll use a simple model that doesn't have all of the nonlinear conductance properties. Okay, very good question.
One more question, chat, but first we'll find the graphs would be a good idea.
Yeah, yeah. All right. So yeah, thanks. Well, take the next question after the break. So guys, we're going to do a 10-minute break. And then we're going to come back here for practical session to finish off the hour. For the practical session, what you need is a web browser and the internet. Okay. So I actually haven't checked about the internet access in this room, but I'm assuming that that's okay.
Access to the U of T Wi-Fi.
It's for U of T at least, yeah.
And Edge.
So when we come back, we'll show you the link that you need to go to, and then that's all you'll need. Okay, so 10 minutes. See you back here at 4:25. Sorry, yeah, 4:30. Okay, guys, let's gonna get back to it. How are we doing with getting ready for following along? Is everybody who wants to ready with the browser open and so on? Yeah. Okay. How can we get you access to the Zoom? What you could do if you can manage it is if you can copy the URL. Yeah. Okay, so you're hopefully getting there soon. Everyone else good? People on the call? Is anyone having any technical issues getting the Colab notebook up and running? Ready from Daphne, thank you. Okay. Okay, so I think we'll kick off. So general question for people first, and show of hands and be honest, please. So who has like zero experience with Google Colab? Okay, 1, 2, just 2. Okay, how about like seen it before but not particularly comfortable? No one. Okay, so like total boss. Yeah, no one's a total boss. I'm a total. Yeah, but reasonably confident. Okay, good. Modesty in the room, respect. Okay. Okay, so I think a few comments for the folks who are new to this way of doing things, and we'll crack on. So who was it? Was you two who are kind of not super familiar with Colab? Right. Okay. So seen Jupyter notebooks before, but not Colab. Same. Okay.
One other person understood as well.
It's the same thing.
Well, they just said need that they don't.
Okay. So you're familiar with Jupyter notebooks, then you're 90% of the way there. Colab is just like Google's platform for Jupyter notebooks. So there's a few things that maybe are kind of orienting things to know about that are slightly different from Jupyter notebooks. One is the keyboard shortcuts. So if you're kind of regular users of Jupyter, then you probably have the, know the keyboard shortcuts. Like that's certainly a good idea. There are keyboard shortcuts in Colab, but they're different. So you need to learn them. So you go to command palette, and then let's see. Yeah, so you see these kind of things on here. So things like insert code cell, right? That's a common thing you want to do.
In Jupyter, that would be like escape A to add a cell above. In Colab, it's control MA, right? So there's a few differences in your keyboard shortcuts that you kind of just need to learn what the equivalent is, or you can, use the buttons if you don't want to learn keyboard shortcuts, but I would recommend you do that. Pretty much everything else is identical to Jupyter notebooks, the Markdown, Python code, and so on. One thing worth knowing about Colab, or maybe two things worth knowing about Colab, that are different and kind of better than ways you're probably used to with Jupyter, is access to files and kind of portability. Okay, so our portability, Colab is everywhere. Like, you have absolutely unrestricted access. All you need is a browser, and you're up and running. And you often have like quite a good amount of kind of free tier level compute before you start, getting crashes and so on. So you can do an awful lot of stuff in Colab without having to, fire up Python on your machine or log into Cynet or do something through a firewall, anything complicated. It's just right there in the browser. So that's great, particularly good for things like tutorials, right? So I've done a lot of tutorials over the years teaching this kind of thing. In the past, what we would do is we would install software on people's computers. And oh my God, installing software on computers is the end of any practical session. You're trying to run a workshop. There's always like two people in the room whose computer like doesn't work because it's weird or for whatever reason. And you spend all your time dealing with troubleshooting, just not even getting started, right? So when you think about it, if you're involved in teaching, education, anything like this, like running workshops, then being able to just go to a browser and then you're there, right? Rather than having to install things and negotiate people's operating system variations is a huge time save, and lets you focus on the content. And it's also, in terms of sharing and so on, if you want to make things available to like anyone in the world without any restriction, or needing to have access to certain files and so on, then Colab is a lot better than that for the most Jupyter equivalents. The other thing that's worth knowing about Colab is file access. So this is kind of next level up, let's say. We wouldn't really be looking at this in this kind of tutorial, but if you use Google Drive, then you can mount your Google Drive in Colab and access files in your Google Drive. So if you have some like data or some arrays that you want to, or some map file or something that you want to kind of do some work with sitting in your G Drive, then you can basically, spin up Colab notebooks and work in your Google Drive as if it was your local file storage system with a Jupyter notebook. So that's very useful for kind of portability. And it's also very useful for things like if you do use large files, then you can, mount large files fully in the cloud without having to transfer any data over any connections that you're, working on on your LAN or anything like that. So that's useful. Okay, so I'll leave it there for general comments. Let's jump in. Props to Parser for this tutorial. This is all Parser's work. So I'm going to defer to him on questions I don't know the answer to as well. But these, what he's done is he's taken some of the main pieces of the lecture and he's implemented them and, picked out some salient points to communicate that we've discussed and, done it in a kind of accessible fashion. I think he's done a really good job. So let's just kind of move through. So we kick off with some imports. I don't need to sign in. What? It's not. Weird. Okay. So we're going to start with the passive membrane stuff. Okay, RC circuit. So we had this equation in the slides, the passive kind of leak current structure for membrane potentials is this simple differential equation on voltage. So we've got a function defining that. What? This is not okay. So execute first cell. Okay, so we're going to define this equation and a simple run function that takes initial condition and some of these parameters. Set the parameters here, define time, initial condition voltage, and these parameter resistance and capacitance parameters. And then what Paz has done is done some nice widgets, interactive widgets, so that we can basically vary these parameters and take a look at how it changes the behavior graphically. So the main thing that we talked about with the with the RC circuit, with the linear property, is like how is just a simple thing really, is like the shape of this curve as a function of the two parameters of the RC circuit, the resistance, the capacitance, and then the input current. Okay. So there's no main point really to make here, other than to say, you know, if you want to understand or explore or kind of think about what the range of behaviors are for these linear passive membrane properties, then you can kind of get there in about a minute by just playing around with these dials and see the different shapes of this curve in terms of like how quickly it asymptotes to the steady state. Now, oh yeah, V equals simulate membrane. Yeah. So the thing that I was saying just now that you missed was just kind of showing some minimal things about the shape of the membrane response, the linear membrane response with these dials. So I'm going to move quickly on from that. The notebook that Parza has a, is this it?
Yeah.
Has a thing for you to fill in to confirm that you're engaged and following, which is this line here. Yep, okay. And that's going to be R times C equals R times C. Yeah, so in the interests of following along, guys, just do what I've done here. So I did R times C on this one, and simulate membrane, which is the function in the previous cell here, just copied over. And here now we can kind of play around with the, right, so okay, so we're looking at the same thing again, the membrane response, the linear membrane response, but just identifying the point at which t equals, sorry, tau equals RC. And that's the number that defines the time constant for the RC circuit. We talked about that in the slides. Yeah, but so RC circuit is basically kind of boring. So onto Hodgkin-Huxley. Run the cells. Here we go. Okay, I'm going to zoom out a bit so you can see a bit more. Okay, so this is version of a figure that we've seen a few times, where we've got the current injection on the bottom, square wave current injection, and then the membrane potential, the voltage trace here at the top. And I'm just going to trace this through different levels of the current amplitude. Okay, so this is like 1 up to, I think, 30 is the highest. So at the lowest level, actually 0. So 2 things we've got here. One is, okay, there's no current injection, so nothing to see here from the point of view of the neural response. However, we've got this thing here at the start. Okay. Now, people who haven't worked much with these kind of simulations might not have seen this before, or if you use kind of simulated tools, they might not show you this part of things. But whenever you do a numerical simulation, whenever you integrate some differential equations, you have to choose some initial conditions, and basically you get some weird thing. Unless you know what the initial conditions are, and you have some specific interpretation of them. But usually in this context, we choose some initial conditions that are like within a plausible range, you know, in this case, like the value of the membrane potential. And then we just run what we call a burn-in, where we say, whatever happens at the start here, I don't care. I'm just going to wait until it's done some basically initial transient. The membrane potential has gone up and down. It's actually behaved a bit like a damped oscillation, but it's returned to baseline here. The point that I care about is after it's returned to baseline. Okay, so now I'm going to start ramping up the input current slowly. So that's happening over here. Okay, so now we put our current injection. If we kind of take it down a little bit, so we have this would be a sub-threshold response because it's not crossing. Yeah, so quite a if you watch the y-axis here, there's a big difference in the magnitude. So we're going from the resting potential -65. We have a current injection, but it's not very strong, 2 milliamps. No, 2 microamps. And sorry, 2 microvolt. No, 2 microamps. Yeah. So we have a response, but this is actually a sub-threshold response because it's not crossing our threshold. Threshold is -50. So, but we'll keep ramping our current up. Okay, and looks like the threshold, the bifurcation, if you like, happened pretty early on in the current ramping here. Here's our action potential. Okay, but just one action potential. So we've got a square wave injection, but it's not a strong enough current injection to produce more than one action potential. He's clearly got close, right? See this recovery here. Probably needed to go a little bit higher, and then we get our second action potential. So if we crank it up, yep, that's exactly what we get. So we've got 2, and then not quite got our third and our fourth, but as you can imagine, and as we saw with the FI curves, we keep cranking up the current. We're going to get more action potentials, more higher firing rate. Okay, so firing rate increasing as a function of the amplitude of a square wave current injection in the Hodgkin-Huxley here. Moving along. FitzHugh-Nagumo. So again, Parser's got a nice little model class here that initializes and then runs the derivative, computes the derivative, and also plots the nullclines or computes the nullclines that are going to be plotted in the phase plane. Nice phase plane plotting function here. And here we go. So, you know, this is what we've seen a version of in the slides before. how are we doing on questions here? Are you seeing?
This is some.
Okay, I think you're seeing it now. Okay, guys, right? Yeah. So here's our phase plane. This is not Morris-Lecar. This is FitzHugh-Nagumo. Okay, but the basic structure is the same. We have our fast variable on the x-axis and our slow recovery variable on the y-axis, and we're gonna do the same thing that we did just now in the Hodgkin-Huxley, which is ramp up the input current and see how it changes the behavior. Okay, now the you can see our phase plane, our phase flows, the arrows in the background here, and our fixed point being plotted more. It's kind of, sorry, that's the initial condition. Yeah. So there's a little line, I think you can see here in green. Yeah. So this is the activity from a starting point over here, and you can see it's dropping onto the fixed point intersection of null lines very quickly there. The input current is essentially, yeah, okay. So now we're changing this parameter, the input current. And what you want to see, I want to show this, is when the intersection of the null lines, whether it occurs on the descending or the ascending limb of the cubic null line, right, the red one. The point where it changes from being on the descending limb to being on the ascending limb is the point at which we get our Hopf bifurcation. Okay, so if we just kind of track this through now. So we're getting fixed point, we're still getting quite kind of damped stable behavior here because it's kind of just moving straight over to the intersection here for this input current level. But okay, yeah, I've gone and I go one step below that. Yeah, okay, so we're still kind of sitting on the fixed point essentially. And I think now we're gonna cross the
Still fixed point, just there's a dip in the beginning and that falls into fixed point.
oh yeah, you're right, thank you, Pazin. What we're looking for is the point where so you can see what's going on here as we kind of crank I up is basically the red null line is just moving upwards. And as I said, so the point where the intersection becomes on the kind of ascending limb, so when the red null line is moving up as opposed to moving down, that's when we start to see the emergence of the limit cycle. And that's the presence of our Hopf bifurcation. So if I'm just going to move through to kind of a really strong point here. So you can see here, this was our initial condition, and then we're moving on to this manifold, and then we're going round and round and round. Okay. And I think, okay, stuff's gone really, really wild if we go higher. Okay, so let's play around with the time scale a little bit as well. Time scale is the rate constant on X, on Y, on W. Yeah, so it's the speed of the recovery variable. Yeah.
It's a scalar, but the derivative of the variable.
Yeah. So I think this is going to basically determine where we have rapid transients or slower transients. Or where's the sweet spot for this? Around here?
It just makes the change steeper with the arrows.
Yeah. Yeah. Okay. So kind of small qualitative changes when we change the time scale there. Okay. I think final point here on the tutorial, and
look at this leaky integrating fire neuron. And again, we're going to ramp up current. We're going to see how it changes firing rate. So that's like the common theme in, I think, more than 50% of everything we've talked about in the lecture so far. And a lot of actual computational neuroscience, right? It's a very easy thing to do experimentally, is inject current. And it's a kind of standard way of characterizing things. So in the case of the LIF model, what we're looking for is the point where we reach the threshold, right? We reach the red line, and then we cross it. And then what the equation does is it just resets us back to our baseline. We drift up again, back to baseline, drift up. And as we saw with the other models as well, right? We kind of increase the current, increases our firing rate. Increases our firing rate. And I think the final point in the ignore the Poisson neuron. The final point, which I won't run this time, but we saw in the slides, is the FI curve for that. So this is, you know, showing that property as well, right? So once we start hitting the threshold point, we get a kind of trend, a linear trend upwards in the firing rate as a function of the input current for the LIF. And similar, if we plotted the Hodgkin-Huxley, we'd see a similar behavior there as well. So that was a really quick run through this notebook. You know, we could spend more time on that, but I think that got to the point. What you'll see in the notebook here, so do kind of take the time to go through this and read, you know, a little bit slower than I went through here. What you'll see in this one, or sorry, a kind of slightly extended version of this one that we'll give the link to, and the other tutorials throughout the course, is some tasks for you to do, some questions and some quizzes, some of which will become the assignment questions that you can answer. And there'll be a little bit more of a kind of journey for you to go on in exploring some of the models. So we'll have some slightly more involved exercises for you as we go. This was a relatively quick and simple first pass. Any questions on, yeah?
Sorry. So why is it all the way up? You don't see a similar sort of.
So you're saying when we go up to here, so why have we not got a limit cycle here? I think we might be going,
Different bifurcation again. So
we've got a fixed point here.
Focus on the intersection between the null points, and that essentially determines the behavior of that stable point. If it's or if it's a stable or unstable point.
Yeah.
So when
So-
it's
So you know, so the phase portrait isn't actually kind of being plotted here, but if you had some arrows around here, you'd see that they're pointing towards the intersection. So there's a stable focus. So yeah, we'd give that with a filled circle.
So do neurons similarly have a between which the correction form would
Yes. I mean, usually the way to answer the question is to actually explore and get a geometric answer. So we could have a go at that. So we've got the limit cycle, okay? And then we're moving our current up. And then the point where it disappears is, here we go there, right? So now we've got a kind of damped oscillation moving down to a fixed point here. So we've moved
we've moved through another bifurcation here from the limit cycle into a presence of a stable fixed point. Yeah, and I guess, is this now because we're on a descending limb again?
Yep.
Yep. Yep. There you go. A question, or are you just hanging? Yeah. Okay. Any other questions, guys? No, nothing burning. Okay. Well, it's been a pleasure. Hopefully, you've enjoyed what you've heard today and found some of it useful. I will post the slides and what we'll do for the exercises, guys, is we're going to put a document in the shared folder that has links to the Colab notebooks rather than dropping the notebooks in themselves, because they're web hosted. So there'll be a document where they'll kind of populate as we go through the weeks with like a link to the next tutorial, and the slides will be up in a few hours as well. And see you guys on the forum. You know, any questions you have from things that we've talked about, or other stuff, please be active. It's a lot more fun if there's some engaged discussion, between you guys, not just kind of with me alone. That's what I'd really like to see over the next few weeks. And looking forward to seeing you guys next Wednesday.